Welcome to the Game Changers podcast, where we explore the stories, strategies, and innovations shaping the future of healthcare and life sciences. I'm your host, Roy Wills, and today we're diving into the most transformative frontiers in biotechnology, gene editing. My guest today is Jack Crawford, CEO of Demeetra, a company pioneering the next generation of gene editing technologies designed for real world impact. Demetra's mission is simple yet ambitious to make gene editing accessible, scalable, and commercially viable across healthcare, biotechnology, and agriculture. In this episode, we'll explore how Demetra is helping global innovators bring genetic breakthroughs from lab to life and leadership vision driving it all. Jack is the founding CEO of Demetra, where he focuses on providing partners with commercially applicable gene editing technologies through simple, accessible licenses. His role includes developing cannabinoid-based therapeutics. Prior to Demetra, he was at Hera Biolabs, where he successfully led a complete business pivot to spearheading the first fully immune deficient gene edited rat models for oncology xenografts. Also, while serving as VP of Business Development at Transposagen, Jack was instrumental in laying the foundation for commercial applications. Jack holds a BS in biochemistry from Virginia Tech and a master's in biotechnology from UPenn. Jack, welcome to the show. Let's get right into it. So let's start with the story. What inspired the creation of Demetra?
Thanks, Roy. It's a pleasure to be here. Really appreciate the opportunity. Demetra is a spin-out of our former gene editing technology company called Transposagen. Transposagen began 15 or 17 years ago, and I started with them early on, actually, myself. And it focused on transposases, which is a type of technology that can integrate genes into the genome for expression. And we still use those. But in 2018, Transposagen sold most of the assets of the company and also had already created a couple spin-outs. And we had some success in applying gene editing technology to drug discovery applications. So most of this is engineering cells or cell lines so that researchers can discover whether a drug target is validated or not, that kind of activity. So we had some success there. And during this period of time, and still today, but especially about 5 years ago, there was a lot of enthusiasm around using gene editing in agriculture. And so we had a small collaboration with the University of Kentucky with some of our technologies. We saw that there was some promise there, and we started. We thought we would create Demetra, solely dedicated to applying our gene editing technologies in agriculture. Now, that was the beginning of the company. Since then, I do want to get into the background and how we pivoted out of focusing on agriculture. It's an area we are still interested in. But I think there was a market fit issue for us and for gene editing in general in agriculture, in terms of starting a standalone company with gene editing technology alone. We had a lot of technical success and a lot of business success during that time we were focusing on agriculture. However, what we realized was the key component of agriculture is what the industry and the breeders called elite varieties of crops like corn, soybeans, strawberries, tomatoes. So these companies have been breeding these crops for decades, if not hundreds of years in most cases, within one company. And they've been studying the traits And you really need access to those lines in order to really get the value of gene editing. So then, if you try to partner with those companies, that's a good strategy.
Hmm.
But what we saw in the bioproduction space, and when I say that, I mean the production of biotherapeutics. These are usually antibodies, proteins, hormone replacement therapies, that kind of thing, vaccines. What we saw is 45% of all the biotherapeutics are made in one cell line. That cell line is called CHO cells. And we could easily get a commercial license from that cell for that cell line and use our editing technology on that. And then we would have a much more streamlined route to market, route to value. And so I made the decision basically at the end of 2024 to pivot and get into biotherapeutics. So sorry about that long explanation, but that's the background.
No, no, that's very helpful, and I think that'll help the audience today to really understand where you started and kind of the direction you're headed into. And I actually have on my genius T-shirt here for you today, so wanted to to give you a little bit of a shout out there. So it very exciting. I mean, so it sounds like Demeetra sits at the intersection of deep science and commercial innovation. So how do you describe your company's mission in your own words?
Thank you. So right now, what we're trying to do, our mission is to provide gene editing technologies and engineered host cell lines to the biotherapeutic production industry. Um, and I believe this is important because we're specifically tailoring our technologies and our cells for that specific industry. So there's a lot of applications of gene editing technology, but we're focusing specifically on that problem. And for the last couple decades, biologics have become the most important drug class, basically. And there's really a new revolution going on in how these antibodies are designed and what they can do. And they're becoming more and more complex. And I know we are going to talk about AI. Everybody wants to talk about AI, but the process of getting to from an idea or a drug target to designing a protein that can possibly be screened or even put into clinical trials is going to go fast, much faster, I think. And some of these molecules are extremely complex. That means they're very, very difficult to express. So for the audience, the way all these molecules, all these medicines are made, most of them are made in cells. And you need to engineer that cell to make these products. And the more complex the product, the more difficult it is to express it. And so, what we're trying to do is we're trying to optimize those cells so we can get higher yield. Our clients can get higher yield, and they can get better quality and not have so much risk of failure. So that's our mission.
That makes a lot of sense, and I have a little bit of experience of working with pharma companies getting drugs through clinical trials. So I assume, from the gene standpoint, it's got to be a little different. So I would assume it's probably a little bit more complex.
Well, it's one piece of the puzzle to get a drug from idea to through the clinical trials and onto the market. I mean, without any, I don't know, 1,000 things going right, it could fail, right? And expressing the molecule at a viable level is one of those things, and so that's what we're trying to help with.
Makes sense. Makes sense. So thank you for that. So take us back to those early days. What were some of the toughest moments building both credibility and capacity in such a fast moving field?
I would say, in the beginning, we were 100% focused on agriculture, like I said. And I had never had any experience engineering plants myself. And so that was basically like starting from scratch. We had the core technologies that we in licensed or acquired from our parent company, Transposogen. But they had only really been used in cells and mammalian applications. So that was getting that to a point where we could demonstrate that this technology could work in plants and could have value in plants was quite difficult. And then the other thing is with a lot of these moving and how fast things are moving was finding that market fit that I mentioned earlier. Um, there's a lot of moving parts in agriculture, whether it's the regulation, what countries are accepting gene edited crops, what countries are not, and then we had to find a market fit. So that was extremely difficult, and I'm glad that we landed where we're at now. Actually,
Excellent, excellent. I'm glad you did as well. So, on a personal note, how was your journey as a scientist or leader influence? How has it influenced the way that you guide Demeetra today?
I would say I am very hands on. I like to get in there and get my hands dirty with what is going on in the lab, in the production process. And I think just like the drug approval process, in our small part of that, any number of things, if one little thing goes wrong, it can make the entire project or the entire process fail. Um, and we've got and we try to hire the smartest and the best people, and they are, I think. But um, I think it's really important for the business leader to go in and take it from a high level and really communicate and say this is the goal, and because you don't want your people optimizing and automating a process that shouldn't exist, right? So I take that to heart. I go in there, I get my hands dirty. I'll give you a couple examples. When we were doing the big plant projects that we were doing, things were getting a little bit slower than I was hoping, and so I asked the scientists and the management what was going on. Had a few meetings. Then finally, I said I got to get in there and take a look. So I went in there, and one thing is they were growing each clone, each engineered plant up to I think 12 or 14 plants to test, and um, I noticed that they were growing them all the way up to maturity, even if they weren't gene edited. And I said, well, why are you doing that? And they thought that um, some of the plants might obtain an edit later on in their life cycle. And what I realized was there was a miscommunication between the people who take care of plants and the molecular biologists who really know what they're doing on the gene editing side. And it doesn't make any sense to grow a non-edited plant all the way up to maturity. So that's one small example. Another thing that more recently happened is we have incubators that hold these cells, and there's a process where they take the cells from one incubator that is at one temperature level and move them to another that's at another temperature level. The problem with that is we have 8 incubators, and this makes it so you can only use like 4 of them at the same time. So I said, have you ever tried just turning the temperature down in one of them? And they said, no, we've never tried that, but it takes like 12 hours. And we think that could disrupt the process. And so we ran the experiment. It had a little bit of an effect, but not enough to really constrain yourself to that small amount. So I like to get in there, never feel like any type of work is beneath me. Always go in. I think people talk about these micro improvements that a company might have. And I
Mhm.
think those all add up in meaningful ways. Um,
Absolutely.
and then the final thing on this is you got to challenge your people, challenge them to do better, but also challenge them for clarity on what's going on. And sometimes that can be kind of uncomfortable. Um, and but you have to be okay with that as a leader, and you have to be okay with any consequences of that challenge. Um, because if a particular person's not okay with a challenge, then they might not be a good fit for your business.
Absolutely. I completely agree. I understand, those parameters as far as a leader, and those are some of the, things that you have to put in place to grow people. So I think that's super important. And it's fascinating how purpose and innovation intersect in your story. So let's unpack what that looks like in action. So how Demetra's cutting edge making science into scalable solutions. Let's talk about that a little bit. For our listeners who may not be familiar, can you explain what makes Demetra's gene editing platform unique and how it differs from the more traditional approaches like a CRISPR?
Sure. So CRISPR-Cas9 is a revolution in genetic engineering, primarily because of the ease and the efficiency of targeting a gene. So it uses a synthetic guide RNA that can, and this is a piece of RNA that can specifically or relatively specifically bind to a target, and it complexes with a protein called Cas9. Cas9 is from bacteria, and it cuts the DNA. And once it cuts the DNA, you either end up with a deletion in that gene, or if you supply it with what we call a donor vector, it can integrate something at that exact site. So that was a huge advancement versus everything that came before it because it was extremely burdensome to design the protein engineering, get these protein binding domains. The problem with CRISPR Cas9 is that it's monomeric. And what that means is once that guide RNA touches and binds to the DNA and it's complex with Cas9, Cas9 will cut at that spot. And so the problem is sometimes guide RNAs bind the wrong spot by accident. So this is called off-target mutations. You don't want off-target mutations, obviously. It can result in instability with for example, these cells that produce these biotherapeutics can become unstable if you have off targets. So what our technology does is it's called Cas-Clover. It uses two guide RNAs that come in at the target site, and they complex with Cas9, but it's an inactivated Cas9. So it just binds to the DNA and does not cut it. And it's fused to what's called a dimeric nuclease called Clo51. The difference between a monomeric protein and a dimeric protein is Clo51 will not cut the genome, will not cut the gene unless both of them are at the target site at the same time. So if one binds another spot by mistake, it doesn't cut that spot.
Mm,
So it significantly reduces the chance of off-target mutations. The other thing I want to touch on is we've actually combined the foundational intellectual property of CRISPR-Cas9 with our Cas-Clover intellectual property, And this, I think, gives us and our clients the strongest commercial freedom to operate in cell line development and bioprocessing. And so that's extremely important and part of that tailored approach.
Makes sense. It sounds like you've applied your technology across pharma, biotech, and ag tech. Can you share an example or a story of a partner that used your platform to solve a real world challenge?
Yeah, actually, this is a great story. I hope it becomes a great commercial business success. We'll see. But it's actually a yeast mosquito biopesticide that we made in collaboration with uh, a researcher at Indiana University, Dr. Molly Scheel. And Roy, do you know the most dangerous? I gave it away, but what's the most dangerous animal on the planet?
I was definitely thinking humans. I was going to say human.
No, it's mosquitoes. Yeah, yeah. So mosquitoes kill like 600,000 people a year,
Really?
and by disease, and all over the world. And actually, some of those diseases are starting with climate change and whatnot, starting to come up into North America, Florida, Texas, California. Um, Malaria and dengue and all these things. And so this researcher discovered a couple of genes that are unique to mosquito. And if you turn them down or turn them off, it kills the mosquito. Um, and these are delivered by what's called silencing RNA. This was a technology that came out right before CRISPR, and the researchers that invented this also got the Nobel Prize. And it's a brilliant technology, and so she wanted to deploy this worldwide, and it's got to be relatively cheap because it's going to be high volume, right? And and I should before I get into that, it's very very specific. So you can feed these to honeybees; it doesn't do anything to them. If your dog ate it, it wouldn't do anything. If you got exposed to it, so it's extremely safe. And the so she had been thinking about producing these in yeasts. So you can have them synthetically made, but it's very expensive. Um, but at the same time, you can use a yeast to produce them at a high level and then grow that up into these in huge fermenters, kind of like a brewery would,
Yeah.
a little bit more controlled than that. But um, and then you've got your product, and it can be deployed everywhere. The nice thing is that mosquitoes are naturally attracted to yeast and they
Mhm.
like to eat it. It's got sugar, it's got carbohydrates, it's got proteins and all that. Smells nasty when it's in, water, everything mosquito likes. And so they'll go and they'll eat it, kills them. So we had a collaboration where we used our gene editing technology to make a super producer stable yeast cell line that
Mhm.
makes these products and actually that makes these shRNAs. And that product was successfully licensed to a company called Anvu, which is was the former environmental protection group for Bayer Crop Sciences. So I know they're developing it right now and testing it in field trials, and I'm excited to see what happens with it. But
Hey,
in addition to that, there's many other things, but that came to mind.
that sounds very exciting. Um, so back on track. So how do AI, data, and automation fit into your process today? Do they help to accelerate your innovation cycle at all?
Yeah, in a couple ways. Now we're just I would say, scratching the surface with AI right now at Demeetra, but we have successfully used it to engineer proteins, which I think one of the biggest um, advancements in AI for biosciences was something, a big collaboration, but a Google AlphaFold. And so this software, after learning from many, many iterations of proteins, can predict the protein structure just on the sequence. This is a huge and reliably do it. That's a huge advancement because before that it would take 4 or 5 years to figure it out. Um, so we have used AI systems that integrate into protein prediction, protein structure, and we've used AI companies that specialize in this area to modify our proteins to make them even better. The gene editing proteins, that is, to make them even more efficient. And a gene or a protein or an mRNA are kind of like language, right? They're sequences that can be read and understood, but there's just vast, billions and trillions amount of combinations. So it's perfect for AI, really. I think um, the other thing that we're using it for is to narrow down to increase the speed of the cycle on product design, I would say, in terms of literature research. Um, and my experience there is that it's a really powerful tool for that. But once you narrow it down and you've got some critical decisions to make, you always have to read the primary literature, the key primary literature. So that's helping us there as well.
Absolutely. No full shortcuts there at all, obviously. So makes sense. So was there a moment recently when you and your team realized you're on to something game changing?
I think when we realized that we had a huge commercial advantage in targeted gene editing for bioprocessing, for this biologics manufacturing space. And that's because of the IP, the freedom to operate, and our know how. And this is relatively new to this space, gene editing, that is. That was a huge game changer event for us, realization for us, and we are building off of that as fast as we can. So I think that's the number one thing that comes to mind.
And that's exciting. It is clear that your model is turning potential into practical value, but scaling that kind of innovation requires collaboration. So let's talk a little bit about how Demetra builds partnerships that last. Demetria's freedom to operate model is a distinctive approach. What inspired that, and how does it change the traditional biotech partnership dynamic?
So freedom to operate, meaning you have your company has a good uh, understanding and pretty confident that you can use a technology for commercial application without having any third party issues, is the biggest challenge in CRISPR gene editing. Um, and that's a whole big story. There were basically two primary inventors of CRISPR Cas. They both patented around the same time, and they've been trying to figure out who actually invented it ever since. But the the fact of the matter is, it's a very popular technology, um, and there are 20,000 patents, and there are probably at least 500 individual companies that have filed patents on CRISPR-Cas9. So, in order to use CRISPR-Cas9 and have clarity around it is a difficult thing to do. Um, so, we set out to specifically use Cas clover as an alternative for this specific space for cell line development, and that's where what drove that decision.
Got it. Got it. So that makes sense now. So, how do you balance protecting your IP with encouraging open innovation among partners?
Yeah, this is a great question. It's near and dear to my heart, and this took some getting used to. So I only try to protect what's core for Demeetra, and not try to get reach through on anything else. So, for example, the sequences of Cas Clover and the sequences of our transposase technology are core. Um, and so when we partner, I only seek to maintain all new discoveries in that space. So if our partner knocks out a gene and they discover something amazing, that's theirs. So we don't have to fight over who owns what, especially if it's unlikely to happen. I've seen that many, many times in these contracts. Um, so we don't fight over the non-core applications, and that really opens up things. And from my standpoint, this actually requires you as the business leader reining in the lawyers, if there are any, in your negotiation, because they don't necessarily understand what's business core. They're just trying to do their job to negotiate the contract and get you as much as you can. Yeah, some people like that, um, but I'm not going to stall or lose a deal for something that I'm never going to use. So that requires a little management there.
No, that makes sense. I think that's a good approach, and obviously it's about getting across the goal line. So I think that's a good strategy. So you've worked with a lot of leading pharmaceutical and ag tech companies. What do you believe makes a great collaboration?
I think people talk a lot about alignment on everything, and I think that's extremely important. But I thought about this question a little bit, and I think that both parties need to want the outcome equally, or at least close to equally. And you can always tell if the other partner doesn't, and it's hard to back out if there's money on the line, for example.
Hmm.
But I think that's the number one key. You got to have your, if it's going to be a big time partnership, you got to both want the deal. Especially if you're a startup or a small company like Demeetra is, it's hard to say no, but sometimes you got to say no.
Yeah, of course, definitely. So technology transfer is often where innovation stalls. What have you learned about making sure your partners succeed once they license your technology?
This is the number one thing. So you, what I've learned is you always have to have somebody that's actually doing the task with their own hands weekly or daily if you can. So somebody that really knows how this works, from soup to nuts, supporting your customers. It's not good enough to write up a technical note and have a representative, a sales rep supporting them, or something like that. Not in this field, at least. I have our people. They talk about bubbles forming in the pipette tip. Like nobody would ever, and how that can disrupt the process, and nobody would ever know. But that the other people on the line are saying, "Oh yeah, yeah, I've had that happen too." You know, so you really need to have somebody. You need to have your group actually working on these things, supporting your customers.
Absolutely, and that makes sense. So, looking ahead, what's next for Demetra's growth? More partnerships, deeper market focus, or something new?
So our deeper market focus is right now we're really focused on engineering a suite of best in class CHO cell lines. That's the platform cell line that can produce these biotherapeutics. And there's different bells and whistles that we can make with the gene editing tools. And so that I would call that a deeper market focus. The other thing we're working on is further evolving our technologies with standard or traditional protein engineering techniques, but also mixing in AI. And then CDMOs, contract development and manufacturing organizations, are a big client of ours. Um, and we are thinking about the idea of partnering on a more deep level with one or more CDMOs because we think we can really bring value to that process, especially if the CDMO does not have the capabilities set up already to do cell line development and engineering. So that's where we're going next.
This seems like a natural progression for you guys, given the time and scale. So, obviously, as we both know, innovation at this level doesn't come without responsibility. So, let's shift to a topic that's increasingly central to biotech, which is ethics and responsible innovation, a place where I've been spending a lot of time recently in my space itself. So, uh, gene editing opens up an incredible amount of possibilities, but also complex ethical questions. How does Demeetra approach that balance between innovation and responsibility?
I think there's a lot of things that can be discussed on this. Luckily, what we do is not super controversial. What we do directly, we engineer a cell. The cell doesn't, is not an animal or anything like that. And that produces hopefully life saving therapeutics. So we don't have to deal with this on a day to day, but we have been in the agriculture space. And I think um, engineering fish for consumption of food or animals, cattle, and that kind of thing. And then, of course, cloning and engineering humans have some very ethical questions. Um, I think biotech and pharma sometimes get a little bit of a bad rap, I would say. But, you know, a good example is during the COVID crisis, where biotech really, I think, kind of came up to the plate and helped out in a very, very good way, whether it be the rapid testing, those are all proteins, and Abbott Pharmaceuticals brought those to the market, or the vaccines themselves. So there's a lot of really good things that gene editing and biotech do, but I think regulating and understanding what does our society want out of this or not is important. Like I think in terms of crop gene editing, and I'll touch on this later, but I think it's important to do whole genome sequencing. If humans are going to consume a product that's been genetically engineered, um, I think that it's important to have the right quality measures. I think whole genome sequencing is a important tool where you can sequence the entire genome and find out exactly what's in there and whether it poses a threat to the environment or the consumer. I think those are all important.
So, as you look to the future, what applications excite you most in healthcare, agriculture, and sustainability?
Um, I think all of them excite me, and I would be interested in working on all of those or many different applications. Um, but gene and cell therapy. I think I had a rough couple of years in terms of failures in the clinic and that sort of thing. But I think what we're coming up, what one thing, this is my hypothesis on that, which I hope to test sometime. Um, that we've got these gene editing technologies for gene therapy and cell therapy. And for the most part, they're a platform technology that can do all sorts of things. And they're trying to be pushed into uh, a gene therapy for a specific disease. So it's kind of like trying to fit a square in a square peg in a round hole sometimes. Um, so with the power of protein engineering now and I think it's not that unreasonable to start with the disease in mind, the gene, whatever needs to be corrected, and create the tool, the therapy, just for that purpose, which would be even more specific and much more likely to succeed. So that's kind of what I'm most excited about.
All right. That is very exciting. So you've clearly built something remarkable at the intersection of science, strategy, and purpose. I want to close by turning the focus to leadership, the human side of driving innovation. Leading a biotech company at this level is both inspiring and demanding. What has leadership taught you about resilience and vision?
So we have a internal saying here, and it's failure is the only path to progress. Um, and I think that kind of sums it up. So it's about humility, um, and it's about understanding that you need to communicate and you need to embrace the failure in order to get the success, and communicate that what happened, why did it happen, instead of specifically not communicating that um, as much. I'm not saying really lying or anything. That doesn't happen very often, but people don't like to talk about it. But we can just like we can learn from all of those things. Um, and then one thing I learned throughout my career is that this is kind of tied into that, but everyone, including me, of course, me, but everyone is wrong sometimes. And given the success rate of startup companies, and especially biotech companies, which is a very low, maybe 5% make it long term or something. There's no right or wrong way to do any of it. So take advice, take counsel from even people that have more experience. Of course, hopefully people that have more experience than you, but it's not always the way to go. Um, and then you'll know when the vision is true and you should proceed and be resilient. You'll know. And if you don't know, then you should not be afraid to pivot.
Yeah, agreed. Agreed. All great principles that I learned in my life as well, especially being an ex-athlete. I think most of those resonate with me. The one that I had to pick up later on was the postmortem, having figuring out why something failed. I think that's critical in evolving and getting better at something or getting to that next level. So I think that's
Exactly.
spot on.
I
Great, great.
actually I make a lot of I know a lot of business leaders do this, but I make a lot of analogies between sports. I wasn't a, I didn't even play sports in college, but I played sports in high school, of course. But like you would go and you run through the tape and you would see, well, you missed this block or you did this or and try to correct that. I think that type of behavior in a business is extremely healthy. So
Absolutely. I, I think you're spot on. So Tell me a little bit about how you keep your team inspired and when the science is challenging and progress can take years in your space.
Yeah, it can take years, but we're all very, very excited to see the next round of results. And hopefully, and those come, weekly or monthly, maybe daily, even some little results. Um, and everybody's kind of on the edge of their seats to see what's going to happen,
Mhm.
and so that keeps us motivated.
Excellent, excellent. So, if we fast forward 10 years, what legacy do you hope Demeetra will leave behind?
Um, we want to be able to say that we helped enable the production of the next generation of biotherapeutics.
Excellent. And just in closing reflection, so just for the audience, what's one common misconception about gene editing you wish more people understood?
So this is more in the agriculture space, but when I was there, I wish, and I still operate in that space a little bit. Um, I wish people understood the difference between gene editing and GMO, genetically modified organism, um, because and why there's that difference. And that goes back to the whole genome sequencing as well. With gene editing, I described this earlier, but you can take this gene editing protein and delete DNA in the genome, and there's nothing else added to the genome. And that you confirm that with whole genome sequencing. That deletion can make a tomato resistant to a devastating virus, or bananas. It can prevent bananas from browning. All sorts of things that could be very, very great for sustainability, for environmental protection, and also for humans. Um, but uh, there's a big difference. GMO gets a very, very bad rap, I think, in this is my opinion, of course, because the very first ones that were made, they were transgenic. And so what that means is you add DNA to the organism. And
Mhm.
what they did was they made the very first GMO, or the first popular ones, were specifically made to be resistant to glyphosate. And so you now have this plant that's resistant to glyphosate or Roundup, and you can spray everything else with Roundup, and everything else dies, but
Mhm.
your plant lives, and that it can produce a ton more yield, right? And that's what it did. Actually, a lot of other pesticides were no longer needed. That's one thing people don't talk about. But I'm not advocating for this, but I think that that's the difference. I think Roundup and glyphosate and that all gets kind of wrapped into gene editing. And I think there's a big, big difference, and I wish people knew more about that.
It is a complicated space, but great point.
Roy,
And
let me. So I met with actually a business collaborator, and they were working on shelf life um, traits, and every day, the Rose Bowl gets filled up with wasted food in the U.S.
so you can create additional shelf life without adding different preservatives and things like that, or
Yeah, yeah. So there's a gene that was discovered, and he was trying to commercialize, that slows down the ripening,
Mhm.
and uh, can extend shelf life like 4, 5, 6 weeks.
Wow.
And then you can get into a lot of ethical questions, right? Not is that good? I think that's good, right? But do the companies want that? If you're selling, I mean, if you're selling bananas
Yeah.
and when your bananas go bad, you need to buy more bananas. But if they don't go bad, you don't need to buy more bananas. So that's another thing.
Absolutely. I mean that's so finally, last question, I promise. And if you weren't leading Demeetra today, what would you be doing instead?
I think I really want to launch that gene cell therapy company. That's where you start with a problem. Um, I think it's probably
Hmm.
gene and cell therapy. Could be agriculture, could be sustainability, could be really anything. And you make the gene editing technology for that problem. That I think is my next step. So
Got it. Got it. I mean that sounds very exciting, and we'll make sure we stay abreast of what you got going on there because it sounds like you've had a phenomenal journey. So, Jack, this has been an incredible conversation, a rare look of into innovation, partnership, and purpose, how that comes together to redefine what's possible in biotechnology. Thank you so much for joining me and sharing Demetra's journey. For our listeners, you can learn more about Demetra and their pioneering work in gene editing at demetra.com. Until next time, I'm Roy Wills. This is the Game Changers Podcast. Stay curious, stay inspired, and keep pushing the boundaries of what's possible. Thank you so much, Jack.
thank you, Roy. I really appreciate it.